While everybody opens, are there any questions from previous lecture? I hope how Illumina sequencing, Solexa sequencing works was clear to everybody. What were the technological innovations? What are the patents involved?
Yes, sir, it's clear.
Okay. All right. So I can see that 6 people have raised their hands out of 9. They are still okay. 7, 8. Jaskeen, can you open the link? Okay, great. All right. Please keep your hands down. So today the idea is to get familiarized with organism-specific databases. Um, so for example, I introduced you to GeneCards, which was an organism-specific database for genes. Today we're going to work on 3 databases: Ensemble, something that I was introduced in the class. Then I'm going to use Ensemble Genomes, which is for bacterial genomes, viral genomes, and lots of other species. And then in the end, we're going to use the UCSC browser, which is sort of, it's like a one browser to rule them all. It's like a very comprehensive tool, not only to visualize data, but also you can get information about how to actually analyze some of the data sets that are out there. So here, there's a short YouTube video which I would like you to see when you are going to the UCSC browser. And in this case, I would like you to save the session that you have created in the UCSC browser and share the session. You have to share a session link in the assignment submission. Okay. And for UCSC browser, there is this specific tutorial document, and there are lots of exercises given in this, but I would specifically request you to go to the exercise 1 to 13. And after you have done
Oh,
that,
sir, we have to save that on YouTube, right?
no. So when you go to YouTube, so this in this browser, you can actually create your own analysis session, and it will create a unique link for each one of you. So I would request you to save that session, right? And paste that unique link here. Once you see the YouTube video, you will be able to understand what I mean by a session. Okay.
Okay, sir, understood.
Yeah. So there are 13 steps, 13 exercises, simple exercises about getting familiarized with the browser. And then you can, for example, also explore the coronavirus genome using the UCSC browser. So that is the next exercise after you have done this. And for any help with any of these steps 1 to 13 that you will be required to do for the assignment, you can go to this website for training and they will be helping you out with it. So first, let's go to the ensemblegenome.org. This is something that I think everybody. So there are 3 questions that I have here for you. So you all know that when you enter ensemble.org, it's going to redirect you to that website. Okay. So I think this is quite straightforward. It has actually redirected us to the nearest mirror. So we are redirected to the Asia mirror. So mirror is basically the same website is copied over many local databases so that typically it would make sense for us to access the database which is closest to us. So that's why we are being redirected to the Asia mirror. Most likely this is hosted in Hong Kong or Singapore, which is where a lot of these things are hosted. Okay, so the first question that you have for today is that let us say you sequenced a human gene which is associated with cancer, and you know a part of the DNA sequence, and mutations in this sequence are specifically associated with colon cancer. Now you do not know which gene this particular DNA sequence belongs to, and the goal is to actually use this ensemble tool BLAST/BLAT and find out the gene that this particular sequence belongs to. Okay, so this is the first question. I'm going to go through over all the questions and then you can start to work at it. The second question is, let us say now you know the name of a gene that you're working with it. So in this case, it's oncostatin M. This is a gene which is involved in regulation of cell growth. It regulates cytokine production. So cytokines are basically inflammatory molecules. So they signal your body's immune system um, to say that there is a foreign invader or there's something wrong with the body. So they should mount an immune response. And this particular gene is also involved in maturation of fetal hepatocytes. So fetal hepatocytes is the liver that is there in the growing fetus. Okay. This OSM protein stimulates proliferation of also Kaposki's sarcoma cells in HIV infected individuals. Okay. So now imagine that you are working with this gene and you are studying the effects of mutation on the protein sequence. All right. So you would like to get the cDNA sequence, the complementary DNA sequence. This is the DNA that is going to be made if you were to convert the RNA molecule of the gene using reverse transcription enzyme to make targeted mutations in the DNA that will code for alternative amino acids. Okay. So again, I would request you to use Ensemble to search for the gene and find the cDNA sequence of the OSM gene. So cDNA sequence, remember, it is the complementary sequence of the RNA molecule that is going to be generated out of the gene. There can be multiple answers here. I'm already giving you a hint. Then the third question is, the human ACE2 gene is involved in COVID-19 infection because the spike protein of the COVID virus goes and binds to the cell using the ACE2 receptor, which is encoded by the ACE2 gene. Mutations in the gene can predict the survival of patients. So if the ACE2 gene is mutated, it is directly going to affect how the COVID virus is going to come and interact with the human cells. You want to design primers to amplify the first exon of the ACE2 gene and understand a bit about the genomic region in which the ACE2 gene is lying. Okay, so using Ensemble, I want you to solve these questions, view and export the sequence of ACE2 gene, paying attention to the boundaries of the ACE2 gene exons, and find the neighboring genes to ACE2. Okay, are these three questions clear? Show of hands, please. I think they are very straightforward, simple
Yes, sir.
questions.
Yes, sir. Like that.
Show of hands. Okay, so, okay, good. Please keep your hands down. So the next thing that we'll be working on is Ensemble Genomes. So Ensemble Genomes is a database of bacteria, viruses, a lot of human pests, and even coronavirus, for example. So you have Ensemble plants, Ensemble metazoans. So metazoans is basically worms and insects. Then you have Ensemble fungi, and then you have Ensemble bacteria. You also have Ensemble protists. So this is things like your malaria causing organism, Plasmodium. You have Dictyostelium, right? So basically everything else which is not there in Ensemble is going to be found here. You also have specifically one for Ensemble COVID-19. Okay, so here the questions that you have to answer, two simple questions. You can answer many questions with this, but one question which is specifically uh, um, important for your degree is the ability to, the first question, this fourth question in this case, is the ability to design new enzymes. So nowadays, most detergents, the expensive detergents specifically, the Surf Excels and the Tides, they are typically have a lipase enzyme which is added to the detergent because the lipase enzyme degrades lipids or the oils. So if you have a oil stain on your cloth, then this lipase enzyme is going to get rid of that stain much faster. So I want you to use the ensemble genomes to find potential lipase enzymes that you can use in the detergents. And once you find three candidate genes or three candidate lipases from different bacteria, so this is specifically from bacteria because it would be easier for you to clone these things in E. coli and express them. right? Biotechnologically, through a bioprocess. So this is what I want you to look at. I want you to find three candidate lipase enzymes found in bacteria and paste their names, any three of them here. Okay, so that is the exercise here. The next exercise is also something very interesting. So let us say that in the very near future you see you are a customs, you're working with customs, okay, and you see that a businessman who was returning from Africa, you seized a bag of fruits from them. So typically whenever you will be going crossing borders between countries, they always ask you in the customs declaration form, are you bringing any fruits or any live animals or anything which is alive from another country, right? So many of times, even some of you who have gone or some of you who will go are not going to declare this, right? So for example, some food items you are not going to declare, but there is some issues with that, right? So for example, this very often happens that fruits have some already uh, the eggs of some insects in them. So let us say you seized a bag of fruits like this from some businessmen who were coming from Africa. It could have been anywhere, any other continent, right? This bag contained a few weird looking mosquitoes, okay? Because the fruits typically have a lot of these eggs, which are, if they have not been washed, and let us say these eggs hatched during the process of the flight and small mosquitoes came up. Okay. The officials did not know about these mosquitoes, what these mosquitoes were. Prior knowledge tells us that Anopheles species, okay, Anopheles species are the common vectors, are these mosquitoes which carry a lot of diseases. Okay, so for example, malaria is carried by, can somebody tell me which Anopheles species carries malaria?
A female.
It's the female, but Anopheles species, which Anopheles species carries malaria? This is something that is taught in class, I think, 8th or ninth. Okay, so search for which species causes malaria. So the question that you have to answer here is, as a customs official, let us say in the future, how would you verify one mosquito species from another? Okay, specifically, you have to answer the question in order to answer this original question. How will you amplify the region, okay, where there could be mutations existing that could be characteristic to a mosquito species, okay? And what will you do after amplifying this particular region to know the mutation status? All right, so this is a very practical problem that I'm giving you. You see some mosquitoes that you want to identify. Now you have to answer, you have to design an assay using the genome sequence of the mosquitoes, right? Do you require the entire genome sequence of the mosquitoes, or only one or few genes can help you in recognizing the different mosquitoes, right? What will you use to recognize mosquitoes? That's another question, right? And then how would you do that? What is the process for doing that? So this is another question. So this is more of a design problem that you should be thinking about. All right. So this is I want to see what thought processes you get. So this is the second set of questions. The third set of questions are to do with the UCSC genome browser. So you go and open this and you will be redirected to the genome browser. All right. And specifically, you can look at the genome browser. Let us say you look at the genomes in the genome browser. When you go here, let us say I go to the human genome. It's going to take some time. And now you see that very interestingly, it shows you the ACE2 gene, because a lot of people must be searching for the ACE2 gene. So it's like buffered, right? It's cached. So the most cached page is ACE2. So it automatically shows you that. So here it is showing you that the ACE2 gene is actually present on the X chromosome. All right. This is the location. It's on the P arm of the X chromosome. So this red line is indicating exactly. Oh, yeah. So this red line is indicating where our gene is lying. Now these things here are the transcripts that are getting made from the ACE2 gene. Okay, specifically this ACE2 gene is coming from, okay, my bad, I think I'm moving this. So you can move around the chromosome. Okay, so this is giving you the transcripts which are getting made from this ACE2 gene, and it also gives you the direction of the ACE2 gene. So ACE2 gene is actually lying on the opposite strand. So therefore it's showing you that the ACE2 gene is pressed going from this direction here to this direction. So it's going from right to left. This is indicated by these arrows. Again, these solid blocks are the exons of the ACE2 gene, and the lines that connect these blocks in between are the introns. So they eventually get removed. So if you specifically look at this, so what you are seeing is this is called a track view of the genome. Okay, I'm zooming out. So you see that this is your chromosome. On the top, you have the scale. Okay, so basically where physically you're lying on the chromosome. Then next set of tracks is showing you the transcripts which are being made. The next track is showing you the gene, the annotation of the complete gene, which is present here. You can also, let me just zoom out here. So right now, let me do a 3x zoom out. So if I will do a 3x zoom out, now this is the ACE2 gene, right? And here you can see this is the annotation of the ACE2 gene itself. Then there are some other genes on the other side. So there is this gene called BMX. This gene is actually lying on the opposite strand of ACE2. So BMX is lying from left side to right side. ACE2 is lying from right side to left side. So they are actually overlapping genes. Similarly, there is a gene called CLTRN. It is going it's also in the same strand as ACE2, and it's going from right side to the left side. If you go over the mouse on the gene, you can actually see some annotation. So there is the ID, and this ID is ENST. So this is basically ensemble transcript ID for for this particular gene. It tells you that is lying on the minus strand, and where your uh, mouse right now is, this is intron 3 of the 5 introns that this gene has. Similarly, let me go to ACE2, for example. If I go here, it shows that I am specifically looking at the transcript ENST followed by the number. This is also lying on the minus strand. And right now I am looking at 11 of the 11 exons that are making up this transcript. Similarly, let me go to this BMX gene. You find the transcript, but this one is lying on the positive strand because the gene orientation is from left to right as shown by the arrows. Now the next track, so just like this, they are layering information in tracks. So whatever analysis that you have done so far, for example, you have looked at the RefSeq database, you have looked at the OMIM database, right? All these things are now laid along the genome in a linear fashion, in a track-wise fashion. So this helps in much better visualization. So this gives you some information. RefC gives you the reference. The basically this is going to tell you that exactly where this gene is. So for example, if I click on it, this is going to expand this particular reference. Okay. Uh, yeah. So now this gives me the annotation from RefSeq. So here you can see NCBI RefSeq genes. It's a curated subset, right? So it's now associated, it's going to give me the IDs associated with RefSeq. So you get ACE2 slash NM underscore blah, blah, blah. So this is the ID associated in RefSeq, right? Similarly, you have OMIM variant. So this basically tells you where in along the genome you have mutations that might be responsible for um, some disease annotation, right? So I can click here and you can find out whether there is some variation or not, some mutants here, but there is no mutation here right now. And I'm going to show you how you can look at these mutations. Similarly, the next thing that you can go here is something very interesting, something that you haven't looked at so far, but I haven't talked about, is the expression of these genes in 54 different human tissues. So for example, if you look at ACE2, let me just like click here. So when I click here, this is showing the expression, that is how many copies of this ACE2 gene are being made across these different 54 different tissues. All right. So specifically, if you would look at this, you see that most of the tissues don't express ACE2. Okay, but ACE2 is expressed at a good level in thyroid, in testes. It's expressed at a good level in small intestine. It's also expressed in your heart, right? Now let's see if it is expressed in the lung or not. It's not expressed in the lung at that good level, but it's expressed in the kidneys, right? So a lot of the actually manifestations that you find in COVID initially were associated with either the small intestine, people were actually getting dysentery. There were also effects in the heart, right? So this is gives you some kind of a indication of where this gene, which body part this gene would be getting expressed. If a gene is getting expressed in a certain body part, that means that gene is most likely having a function in that body part, all right? Or it's able to do the function in that body part. Is this clear to everybody? This is a lot of information that you are sort of getting in a very small area. The other thing is this track is called encode candidate cis regulatory elements. So this is basically just like you have transcription factors, binding sites, there are a lot of other kinds of proteins which bind to DNA to modulate it. These things are together called cis regulatory elements. Cis basically means the fact that these elements or these motifs are lying right next to the gene where which these are sort of regulating. Okay, so this is they are cis to the gene, so they are just next to the gene. So you see all these different rectangles, right? This vertical rectangles, they are actually annotated some kind of regulatory motifs that are there. So let me just click on one of these motifs. So see, if I roll over here, it says this is a promoter like sequence. So that basically means this sequence it can act like a promoter. So let me go to one of these blue ones. This is a CTCF. So CTCF is another DNA binding protein, which is playing a role in changing the physical organization of DNA inside a cell. So let me go to some other one of these brown ones. So this is a distal enhancer like signature. So it's basically saying that this particular DNA part, this region, is working like um, another regulatory motif. So in this case, this regulatory motif is called an enhancer. So just like you have a transcription factor binding site, you also have enhancers. So enhancers are, just to cut a long story short, they are some kind of super regulators. So transcription factor is typically regulating, the binding site is regulating only one gene next to it, but an enhancer can regulate multiple genes next to it. Okay, so this one enhancer might be regulating this entire region. All right. The next thing that you find here is another layer that you are finding information is histone marks. So histone marks are basically modification on the histone tails. So I hope everybody knows about histone tails and how they are modified.
Yes, sir.
Okay. Only Ratnesh said yes. Everybody else?
So we know what histones are, but I'm not sure what histone tails are.
So,
Yes, sir.
so you don't know what histone tails are? Nobody knows what histone tails are? So everybody knows what histones are, right?
Yes, sir, we
Yes,
know
sir.
what histones are.
So histones are highly positively charged proteins around which DNA molecules wrap so that they can be condensed. All right. Now these histones, they are sort of modular proteins, but the N terminus of these histones are unstructured and they actually, stick out like a hair. Like if you have hair on your head, if you comb your hair, but sometimes one hair just sticks out. So histone tails also stick out like that. Okay. And these tails are typically their charge distribution can be modified. So for example, here H3K27 means that the histone 3 H3 at position 27 it has a K. So can somebody tell me what is a K? What amino acid is a K? This is something that might be
Lysine.
asked in your quiz as well.
So is it lysine?
It's a lysine, yes.
So
So
are
can
we
some
supposed to remember the codes of every amino acid?
you should, I mean, as a
Yes.
you should be able to, Ratnesh, it's not a question for you, no, it's for me. You should at least be able to quickly refer to the table. But if you want to continue in biology, these things, it's only 20 letters. Um, yeah, so I'm not going to, I don't mind if you Google it also, but you will get a couple of questions around these these amino acids. I would, the way to do it would be to do it in some form of a mnemonic. So you should be specifically, when we are talking about DNA, you would want to remember about positively charged nucleic acids, amino acids. So arginine and lysine are two positively charged nucleic acids, which are there on the, sorry, no, amino acids again, I'm sorry, which are there on the tail of the histones. And because they are positively charged, they actually further wrap around the DNA, which is negatively charged, and can modulate how tightly or loosely DNA is bound. Now, what is specifically happening here is there is an acetyl group which is added to the this lysine, right, which is there on the tail. All right. Now, acetylation. What does acetylation mean chemically? What is the chemistry? What is the chemical formula for acetyl group?
CH3COO.
CH3COO.
Yes, sir. Negative with a negative.
It's negatively charged, right? So it's, if you're adding a negative charge to something which is positively charged, you're essentially net reducing its positiveness, right? So when you reduce the positiveness, that essentially means now the DNA strand, which was DNA backbone, which is very negatively charged, which was first very tightly bound to the tail, that was positively charged, but now that binding might not be very strong because it's like charges, right? So they are repelling. So when you have things like that,
No, sir, like I made a mistake. It's actually CH3CO. CH3COO is acetate, not acetate.
yeah. So it's, but it's negatively charged, right?
So I guess it's a like radical, or it's not actually charged if I'm not wrong.
Its acetyl group is, I think, CH2CH3.
Yes, sir. So CH3CO.
Yeah, so it's CO, right? So it's got a oxygen, right? So it has
Yes, sir.
it has electrons to give. So
Yes, sir.
so now when it has does that, that basically means that the DNA around this region is more loosely wrapped, and typically around the promoter region, you see that the DNA is more loosely wrapped. That's how the cell is able to identify promoter regions, because it wants the DNA to be loosely wrapped so that your transcription factors can come and bind, your polymerases, RNA polymerases can come and bind and do the transcription process. Okay. So this is one more information which is given. So just like that, you can actually layer, you will find if you go below, you can actually add more and more layers here. Okay, more and more layers, and you can get more and more information. So right now you see that we have selected encode uh, cis regulatory modules, cis regulatory CGEs, right? We have expression, so you have GTEx expression, so this is gene expression across tissues is the short form, the full form of GTEx. So this is what you were seeing earlier. So let us say all these other information is currently being hidden. So let us say I click here and I ask it to show CPG islands. So CPG islands are locations where you have very high density of CGs. So these are called CPG islands. So when I select this, what's going to happen is we are going to have one more layer of information added. So this one more layer of information, let's see, where is it? So you have S3 methylation, you have, so this basically gives you a score of, this track is giving you a score of base pair wise conservation across 100 vertebrate species. And you can see these species here. So for example, this is the rhesus monkey, this is mouse, this dog, elephant, chicken. So the darker the color, the more conservation you have. So for example, these peaks tell you that this particular base pair is highly conserved, and you're going to see some pattern. Wherever you have an exon, you see there is more conservation. Whenever you have an intron, there is less conservation. You can think about why that is the case. Um, for some reason, it is not showing. Let me see. CPG island. Was it able to show? Huh. It's not able to show this here.
So it is visible in green. It says CPG islands. Islands less than 300 bases are light green.
Oh yeah, right there. Exactly. So here you see that this is a CPG, right? So this is it's light green. So maybe I can zoom out and you will see this a little bit better. See, this is all of it is dense now, and you see these are CPG islands shown here. So for all these genes, you can look at gene expression now. These are all the regulatory elements that are there. Again, this is the histone modifications. This is the conservation across. This is conservation for different organisms, right? So typically you will see that genes which are conserved are going to have more darker score, and this is conservation with respect to human gene. So therefore you see that the rhesus monkey is almost identical, right? Because it's in the it's very similar to the human genome. Mouse is also quite similar. Dog is also quite similar. But if you go to chicken, the conservation becomes less. If you go to um, Xenopus tropicalis, which is the frog, also a model organism, this becomes even lesser. And then you have the zebrafish, it also becomes much lesser. All right, so this is a lot of information that you can see in this layered format. You can layer one over the other and you can browse all this information simultaneously. Now, there is a lot of public information which is out here, but as you will see in one of the exercises in the advanced exercises, if you are interested in that, so after the 13th step, you can go and see that I think in the 14th, or I can just open this. So there are different steps here. So the step one is open the browser and see different things. Step two is click the genome browser to left hand side bar. So you see the gateway page where you are. Then you can examine the genome annotation track. Something these things I've already done for you, right? You can display different tracks. You can click on these different tracks and explore them. You can put your mouse over them, zoom in, zoom out. You can look at different genes. You can look at some information about these different genes. You can then click and understand what these different colors mean for the particular gene. So this is some information that you will be getting, right? So there are these 13 things. Now beyond that, you can even do some analysis and you can actually add your own custom tracks. So for example, if you did some experiment for say a protein that is binding across the human DNA, you find a new protein which binds to DNA, you can make a track out of that and you can put it there and you can visualize it along with the entire human genome. Or for example, if you were to look for enrichment of 9 mers in a 500 base pair window, you can actually put that information and visualize it in this browser. Okay, a lot of this information which is out here. So let me just click on one of this. So let us say I'll click on the CPG island one. So let me just click on this. So this specifically gives you where this location of this island is, what is the size of this feature. So this is called a feature. This tells you the count of CPGs here. This 35 counts, right? Percentage of CPGs, percentage of CGs, right? The ratio of observed to expected CPGs. So it's 81 observed by expectation. So it's very high, right? And tells you some information about how, what this information, what is the underlying way in which you are showing this information. Now, specifically, you can also go to what's called the table browser. And when you go to the table browser, you can actually download a lot of this information in a text format. And the text format that you will be specifically getting this information is called the bed format. All right. All this is given in this tutorial. I request you to please go over it. And yeah, if you have any questions, please, you can always, I'm here to ask. So for example, you can ask for the just genome position from chromosome for chromosome X from 755 to this particular location. You give me uh, all the fields from the selected table, right? And just get the output. So this produces a file like this. Okay. It is giving you all the different kinds of information which is being displayed. So you can specifically here, I gave a very large location. You can even get much smaller information for this. So you can specifically say you want location only these locations. Or you can just upload your own file. So this is a table separated format, tab format. You can submit it. Okay, it's a defined region. And now you can get output. So only for this particular region, you're getting all the information. All right. Is this clear? Some ideas, some basic idea you're getting. Other things you will can once
Yes,
you
sir.
explore, you will start to get more information about it.
Yes, sir.
Okay. So um, again, so this is what you have to do. These 13 steps. It's not a lot. And then I please, because everybody is suffering from this, and you are studying this in class, so go through the coronavirus genome browser and see what kind of information is given in the browser. Okay. Some of the questions again will be asked from this particular part of your ability to explore the coronavirus genome properly and the human genome as well, of course. All right. So I'm muting myself. If you have any questions, please ask me and I will unmute myself and answer.
Okay, sir.